Hello everyone. So in today's topic, I have taken a question where I'll be discussing about the RNA transcription and the protein formation, um, and about alternative splicing as well. So this question I have taken from 2014 life sciences paper from biochemistry section. This is question number 18. So this question stated as the a human gene has only 3 exons, 1, 2, 3 in the given order. So total RNA was isolated from cultured human kidney cells and reverse transcribed. So the resultant cDNA was used as a template in a PCR reaction containing a forward primer specific to exon 1 and reverse primer specific to exon 3. When the PCR product was analyzed by gel electrophoresis, two bands were observed of sizes 2.5 kb and 1 kb. However, when northern blotting was performed, uh, with the same total RNA using radio-labeled probe specific to exon 2, only one band was observed. Based on these observations, which one of the following statement is false? So, uh, here they are trying to amplify one particular gene. So, So for that, they have mentioned that this human gene contains exon and introns. Okay, that signal you can get from the statement that this gene contains uh, 3 exons. Okay, so if it is a single gene, there is uh, if it is a single gene, there will be total 1 exon. If it is having introns in between, then only is having 3 different exon because of the presence of introns in between. So if we'll talk about exon and intron, so introns are the non-coding part, we can say, that doesn't code for anything, and these needs to be removed for a proper functional protein formation and functional RNA. Okay, so yeah, functional RNA. So either you have a setup like, if this is one your gene of interest, okay, this can transcribed as mRNA, and that RNA can form a protein. Okay, but if it carries intron in it, that means this is the gene of, so intron is present and there are 2 exons. So firstly, it forms, let's draw like this, a pre-mRNA that will be spliced. So the removal of intron is called as splicing, and after the splicing we get this functional RNA that is further will be transcribed as a protein. Okay, so this forms the functional protein. So this is exon and this is exon with the intron. So where in between the gene the intron is present, there comes one more steps, that is called splicing. Okay, so suppose if in your particular gene, so what we do if we want to study a gene or anything, what we do, we take the entire genomic DNA part, okay, and from there, if our gene is present, we give the primer specific to that gene and amplify it using PCR and get many copies of our gene of interest from there. Okay, and that we further use for clone it in a vector and then use in our study. Okay, okay, but if your gene of interest harbors intron also, so from here So here this is one particular gene, but it also carries the intron, that if you amplify like this, then you will contain the intron as well. Okay, if you amplify like this, so you will have intron also. So this you will get as a PCR product and containing the intron, and this cannot make a functional protein. If you want to study this thing in a bacterial system or a system where the splicing doesn't work, so this will not make a functional protein. That is why you cannot take it as it is from the genomic DNA. So what you have to do here, that you have to take uh, or amplify such a thing that is devoid of the introns. So that how can happen? So inside the genome, okay, inside the genome itself, okay, or the organism itself, when this gene is getting uh, transcribed in form of RNA, okay, so that is called pre-mRNA. Okay, so when this pre-mRNA makes the spliced mRNA, this particular portion you can take, okay, this RNA portion, and this you can convert from RNA to DNA, okay, from RNA to the DNA that now contains the exons only, not the intron. Okay, so this DNA, so from RNA to DNA conversion, this process is called as reverse transcription. This process is called reverse transcription. Okay, because from this is the DNA. From DNA we get the RNA using transcription. So if from RNA you are again making DNA, that is called reverse transcription, and that DNA will be called as complementary DNA or cDNA, because that will be in complementary of the RNA sequence. Okay, so that is the cDNA that you need, so that you can amplify using the PCR, and then you can clone in the desired vector and use it for the studies. Okay, so this is what you need to do when you have an intron present in your gene. So that is what they are doing here. So in their study they have shown that this gene has 3 exons, 1 2 3, and total RNA they have isolated. Okay, the one which I have mentioned here, they have isolated the total RNA from the cultured human kidney. So suppose we have read about that uh, differential gene expression. So all the parts of our body contains the same genome, but some genes that express in particular organs only. So suppose this particular gene expresses only in kidney, that is why they cultured the human kidney, and from there they have isolated the RNA. Okay, so they have isolated the RNA from the kidney cells and performed the reverse transcription. That means they have made the cDNA as well, and the resulted cDNA, yeah, here it mentioned the resulted cDNA they used as a template in a PCR reaction containing a forward primer specific to exon 1 and reverse primer specific to exon 3. Okay, and when the PCR product was analyzed, the two bands were observed. So how is this happening? So okay, so there is one phenomena. Now we read about splicing. This is the thing which I am telling you. This is called alternative splicing. So in alternative splicing, so there are, here you can see exon 1, exon 2, exon 3, exon 4, and all this is intron, this is intron, this is intron. So 4 exons divided by 3 different introns. So splicing can take place in either way. So all one way, which is this one. So here they have mentioned all the introns being removed and all the exons are joined together in one line, and they are making this functional protein where all the 4 exons are being packed. Okay, in the second one, here the splicing, you can see only 1, 3 and 4 fragment. So the in the splicing process, what has happened, this entire chunk has been removed. So along with intron, this exon portion is also removed, and now the mature mRNA looks like this, 1, 3 and 4. So now this will make another functional protein that might have a different function or similar accessory function, but these are, this protein and this protein are two different proteins now because of the different uh, uh, sequence and structure and might be function. Okay, similar way in the fourth one you can see 1, 2, 3 are in continuation, and this particular portion is being removed, and along with these introns, and these three are present and they are making another protein. So that is called alternative splicing, same gene sequence but coding for different protein because of the alternative splicing. Okay, so if in our question they have given us something like, so in our question they have given us something like this. So okay, okay, so they have given exon 1, exon 2 and exon 3. Okay, so this is what they have given. Okay, so they are performing when they are performing the they are isolating the RNA and making the cDNA, what they are getting on a DNA gel, they are getting two different bands. Okay, two different bands they are getting. So their expected outcome is this, right? Exon 3 to 1. Forward primer specific to exon 1, reverse primer specific to exon 3. So this should give one single PCR product, but they are getting two different things. Okay, so two different things, that means whatever product they are getting, it must contain exon 1 and exon 3 because the primer is specific to those exons. Okay, so there are chances that via the alternative splicing this portion is being removed. Okay, that is why if they have getting some kind of sequence like this, that is why when they are performing the PCR using the primers like specific to exon 1 and 3, then they are getting two different kind of PCR product, one corresponding to 2.5 kb, another corresponding to 1 kb, 1 kb I guess. Yeah, so that is what they are getting here. So, and further they also perform the western northern blot analysis. Okay, so they have performed the northern blot analysis. So in the northern blot, what they have done in the northern blot analysis is being performed. So in the northern blot analysis which they have performed, the same total RNA used as a radiolabeled probe with specific to exon 2, and only one band was observed. Based on these observations, which on the following statement is false. So in northern blot we run the entire RNA, total RNA isolated there. Okay, so there also when they have probed with the probe that is specific to exon 2, so there they are getting only one band. Okay, only one band in the northern analysis. That means exon 2 is present only at one place. Okay, so according to that we can hypothesize that the gene gets undergo alternative splicing. Okay, this RNA undergo alternative splicing, and then why we are amplifying, you are getting two bands where exon 2 specific primary is not there. Because of that you are getting two bands which are specific, getting amplified with the primers of exon 1 and exon 3. And also in the northern blot specific probe specific to exon 2 is showing one band, that is only one RNA is present that harbors the exon 2. Okay, now let's see their statements. So here first statement is northern blotting with a probe specific to exon 3 will show two bands. Okay, so if the total RNA is ran here and the probe that been used is specific to exon 3 here, so it would have recognized both these RNA. That is why it would have given two bands. Okay, two bands it would have given. So that is what the statement says, that northern blotting with a probe specific to exon 3 will show two band. That is the correct statement. Okay, so the gene codes for two mRNA splice variants. So by our detailed analysis of the experiment, we can say that yes, this gene codes for two mRNA splice variants that are being generated using alternative splicing. If the forward primer, so third option is if the forward primer is specific to exon 2, two bands will be observed. Is it? So here they have used the forward primer specific to exon 1. So if it would have been specific to exon 2, here this one would have given a PCR product, but here if the forward primer is specific to exon 2, it would have not bind, only reverse primer would have bound here, and only with one primer the PCR amplification cannot be possible. So if the forward primer was specific to exon 2, it would have given only one band, not two. So that is why this particular sentence is the incorrect sentence. And the fourth option is exon 2 is 1.5 kb in size. Yeah, so here we are getting 2.5 and 1 kb. So entire length of this is, we can say 2.5 kb, and entire length of this you can say 1 kb. So in between these two we have exon 2 missing. So if we'll subtract this fragment size from this fragment size, we'll get the length of exon 2, that is 1.5 kb. So the fourth sentence is also correct. So altogether we can say that this third statement is the false one. So according to their question that they have framed, the correct option is the option C. Okay, this is the correct option. Okay, this particular one, and other three options are the true statement, and this one is the false statement. So according to that you have to answer, you have to identify the false statement. Out of these four, the third statement is the false statement. Okay, it's clear. And this is my NPTEL reference. Thank you for watching.